Hi doctor friends, welcome to my YouTube channel PG Dr. Sindranju. How are you friends? Today's topic is the most important topic, Biochemistry online part first. This is for you and for your best performance in the all types of medical PG exams. So friends, watch this video till last and don't forget to subscribe, like, comment, share, and all my videos are very hot for PG exams. Let us start the HMP shunt. About the HMP shunt, what is the true? True is that occurs in the cytosol, no ATP production in the cycle, and the active in the adipose tissue, liver, and gonad. HMP shunt, also called as pentose phosphate pathway. This is important topic and also known as phosphogluconate pathway, and it is parallel to glycolysis, but the anabolic effect rather than catabolic. It is anabolic in nature. It generates, first of all, NADPH and ribose 5-phosphate and pentoses. So three things it form: NADPH, ribose 5-phosphate and pentose, and its main role in the RBC. It has two steps. A step first for the also called first step is the oxidation, and second non-oxidation. In first, here it is 4-NADPH formation, and second non-oxidation phase for synthesis of pentose and ribose 5-phosphate. No ATP formation takes place in the HMP shunt. Here the second one-liner is within the RBC. Hypoxia stimulates glycolysis by regulating pathway is the hypoxia stimulates release of all glycolytic enzymes from the band 3 on the RBC membrane. Next is the dinitrophenol, also 2,4-dinitrophenol. Its name is 2,4-dinitrophenol, which works as the uncoupler. And the main question is asked, what is the uncoupler? Uncoupler is a compound that delinks the electron transport from oxidative phosphorylation. That means it is for ATP synthesis. ATP synthetic phase is known as oxidative phosphorylation, and it allows oxidation of substrate without ATP formation. So if ATP is not formed by de-link the chain of the electron transport. Uncoupler works like it increases the permeability of inner mitochondrial membrane to the proton. As a result, is that the ATP synthesis does not occur, and energy linked with transport of the electron is dissipated as heat. And uncoupler, it allow the uncoupler allow, that means often accelerate the rate of reaction, that means the oxidation of the substrate via the NADH or FADH2 without ATP formation. The example of such are as the chemicals is like the dinitrophenol, dinitroglycerol, and pentachlorophenol. And another example is that is FCCP, that means trifluorocarbonyl cyanide phenylhydrazone. This is 100 times potent and 100 times more effective than dinitrophenol. And high dose, also high dose of aspirin, it also works as uncoupler. And the physiological uncoupler is the already know thermogenin, thyroxine, and long chain free fatty acid, and some antibiotics like valinomycin and nigercin. These are works as uncoupler, that means no ATP formation but it produces heat, so known as uncoupler. Next is the synthesis of triacylglycerol in the adipose tissue. The true is the synthesis from dihydroxyacetone phosphate enzyme glycerol 3-phosphate dehydrogenase play important role in the phosphatidate is the hydrolyzed. This is important. Next is the proteoglycan. The proteoglycan, what is the true? The chondroitin sulfate is the proteoglycan. They are made up of sugar and amino acid and they carry charges. The proteoglycan, here proteoglycan also called as the mucoprotein or also mucoid. and due to the core protein, the core protein is covalently attached with glycosaminoglycan GAG, with serine is the point of attachment, and amino acid, you have to remember, serine is the point of attachment. Here important thing is that the proteoglycan, here the carbohydrate part is approx to 95% and protein part is approx to 5%. When the carbohydrate part is more than 4%, this is then it is called as the glycoprotein. Okay. Next thing is that what is the role of the proteoglycan or glycoprotein, may called as an enzyme, hormone, transport protein, structural protein. The carbohydrate, what is the carbohydrate in the glycoprotein? Then you may ask that the carbohydrate is the mannose, galactose, N-acetylglucosamine, xylose, N-acetylnuraminic acid. This is the component of the glycoprotein, and the main thing is it is carbohydrate part. And some antifreeze glycoprotein, the antifreeze glycoprotein, in that the tripeptide is the alanine and threonine is the tripeptide present in that antifreeze glycoprotein. It is the 50 repeating unit of this tripeptide which are present in the Antarctic phase, so that it can withstand the temperature below -2 degrees C. And some glycoprotein and which measure function like the collagen. The collagen is the structural function, and ceruloplasmin, already know it is a transport protein. It is transport protein. And immunoglobulin acts as a defense against infection. Okay, defensive function. And thyrotropin, erythropoietin, as a hormonal function. And some intrinsic factor. The intrinsic factor, you already know that intrinsic factor is responsible for the vitamin B12 absorption. And already know the fibrinogen for blood clotting. I have already told that the here the glycan part, sorry, here the glycan part is the carbohydrate part. That means mainly it is glucosaminoglycan, maybe chondroitin sulfate, hyaluronic acid, heparin, dermatan sulfate, creatine sulfate. So here the chondroitin sulfate is a proteoglycan. Also, hyaluronic acid is a proteoglycan, heparin is a proteoglycan, dermatan sulfate proteoglycan, creatine sulfate proteoglycan. So these are, they are made up of the sugar, and amino acid, they carry the charges. Next is that the protein purification and the separation can be done by chromatography, centrifugation, and electrophoresis. It is asked in the AIIMS PG and All India Medical PG Exam. So NEET PG, DNB. So this is important. Also FMGE. So it is important topic. These are all one-liner are important for you in the future exam. So make it note so that you can easily revise. Next is the study of multiplication of the protein in the disease is process is known as the proteomics, and the protein that precipitate on heating to 45 degrees and resolve on the boiling is the Bence-Jones protein. You already know multiple myeloma in which Bence-Jones protein are found. The main important things is that here the northern blot is used for separation of mRNA. Many times repeat about the blotting. So this is very important. Now I am making you clear that the blotting is the, if you make the map, it is east, west, north and south. So you have to remember east is not present, only three things is western, northern and southern blotting. So start with, you have to remember the correct process. That means you have first remember, start with east is one, first, second, third and fourth. So you have to remember by this sequence, the it is second, north is third and south is fourth. So you may remember it by the fourth number is southern, so fourth letter is D, so it will be DNA. Okay, if you remember that, the first have to remember west, then north, then south. The western blot for the starting, and it is protein, and northern due to three things, west, north, south. Middle A is northern, so mRNA for that. And if you want to talk another way, the trick is that the western, northern and southern blot, it is for protein, it is for mRNA, and southern for the DNA. Here in the western, you have to remember in the sequence WNS, west, north, south, and P stand for the primary, and M for middle, and D for delay. So primary protein, middle mRNA, and delay DNA. Another one is that the C terminal end of androgen receptor is concerned with the ligand binding. Next is the technique used for detection of the variation in the DNA sequence and gene expression. It is the microarray. Next is the test used for detection of a specific aneuploid. It is phase, RT-PCR, and QF-PCR. The prenatal diagnosis of hemophilia is best done by PCR, and enzyme involved in the oxidation reduction reaction are dehydrogenase, oxygenase, and peroxidase. Least polar side chain is the methyl. Always remember. Next is the statement true about the facilitated diffusion. Facilitated diffusion. The diffusion is the facilitated that require carrier protein, and it doesn't require ATP. Diffusion is simple diffusion, facilitated diffusion, and active diffusion. When active word is used, that means it requires ATP for the transport, and it is against the concentration gradient. Simple diffusion, it is according to concentration gradient, and facilitated diffusion, it requires carrier protein. Next is the true about lipoprotein lipase. Lipoprotein lipase, it found in the adipose tissue, found in the myocytes, and the deficiency leads to hypertriglycerolemia. The gap between the segment of the DNA on the lagging strand produced by restriction enzyme, that means restriction endonuclease, are sealed by. When sealing word is used, that means it is ligase. When breaking word is used, then it is gyrase. Next is the normal role of microRNAs, the gene regulation. The sickling in the HBS disease is primarily caused by decreased solubility. True about recommended dietary allowance is the RDA. That means recommended dietary allowance is the statistically defined as the 2 standard deviation above estimated average requirement. The plant component not fermented or not degraded by the GI tract microorganism is the lignin. This is also many times repeated in AIIMS PG, NEET PG, FMGE, and DNB. So make it clear. Next is a 10-year-old child with aggressive behavior and poor concentration is bout with PC of the joint, pain, reduced urinary output, and self-mutilating behavior. That means his finger cutting. Deficiency enzyme is the HGPRTase. The false about the lambda phase. Lambda phase, both lytic and lysogenic phase occurs together, it can't be possible. The initiation of visual impulse is associated with the photoisomerization and hydrolysis of visual purple. The metabolic pathway generating ATP is the glycolysis, TCA cycle, fatty acid oxidation. In which tissue glycogen is incapable of contributing to the blood, glucose is the muscles, and the phosphorylase is maintained in an inactive state by the insulin. When insulin is absent, then phosphorylase get activated. The group of protein assisting in the folding of other protein is the chaperone. Protein are shortened by Golgi body, and trypsin inhibit alpha 1 antitrypsin, beta 1 antiproteinase, and egg white. The true about apoprotein. The apoprotein A1 activates ALCAT, apoprotein C1 activate lipoprotein lipase, and apoprotein C2 activate lipoprotein lipase. The types of hypertriglyceridemia associated with increased VLDL and chylomicron remnant is type 3rd. Okazaki fragment is formed during the synthesis of double-stranded DNA, and water-soluble vitamin is the folic acid. All B complex are the water-soluble. Only water-insoluble is A, D, E, and K. These are water-insoluble vitamin, and all other vitamins are water-soluble. These are fat-soluble vitamins. The vitamin required for the carboxylation of the clotting factor is the vitamin K. It requires factor 2, 7, 9, and 10. You must remember. The active form of the calcium is the ionized calcium, and neutral molecules are transported by simple diffusion, and the true regarding the oxygen is incorporated to atom of oxygen, incorporated one atom of the oxygen and required for the hydroxylation of the steroid. The next is the treatment of multiple carboxylase deficiency is the biotin. The basement membrane degeneration is mediated by the metalloproteinase. The enzyme stable at acidic pH is the pepsin. Due to the gastric pH is varied between the 1 to 3, so in the acidic environment the pepsin works better. The method used for the protein estimation is the bromocresol green method, biurate method, Lur method. This is important thing I have making it clear, and that thing I live is very important also, but I will upload that video in the later. Thanks friends for watching my video. Something write in my comments box regarding this video so that I will upload the other video regarding other subject. And thanks a lot for watching my video. Have a nice day.